Hello friends, in this video I am going to explain you about the movement of lipids within the biological membrane. So let's see, consider it is a biological membrane. We know that these are the lipid molecules. This is the head of the lipid and this is the tail, two fatty acyl tail of the lipids, and heads are actually hydrophilic in nature, hydrophilic and tails are actually hydrophobic in nature. Tails are actually hydrophobic in nature, and these are the tails. These are actually integral proteins which are present within the biological membrane. Here is another protein. This is another peripheral protein. So there are actually four types of movements in lipids. Number one is the lateral movement. Second is the flip-flop. Movement. Third is the rotational. And four is the flexus. These are actually four types of movements which are we find within the biological movement of lipids. First is the lateral movement. In lateral movement, actually the lipids, for example, see these lipids actually transfer from this position to any position within the leaflet. Okay, and we know that plasma membrane actually made of two leaflets. This is one leaflet and here is another leaflet. This is called cytosolic if it is a cytosol, and this one is the exoplasmic leaflet. So in case of lateral movement, these lipids may transfer any position within its leaflet. Okay, for example, this lipid may transfer to here, or maybe to here, or this lipids to here. These kinds of movement actually known as lateral movement of lipids. And then this is the lateral rotational movement. Actually, let's say this is one lipid here, this lipid maybe actually it rotate its own axis. It never changes position. The tail remain its place, just it's rotate with inside, either side or either this side. Okay, this is the rotational. And in flexus type, actually lipids remain its place. Let's see, these lipids remain its place, just their tail slight rotate. They actually not rotating the lipids, they just look like rotating, but they remain in their place. Okay, and then the last one is the flip-flop rotation. In flip-flop rotation, actually it is happen very low in number. Actually like see, flip-flop movement means lipids from one leaflet to another leaflet. From, for example, see, this lipids is here, but it is actually not happen regularly, because we know that this cytosolic tail, these tails, lipid tails, are actually interact with each other by hydrophobic interaction and van der Waals interactions. So these two interaction, for movement, they need to first break these two interaction, then they need to pass through very hydrophobic region, this, then they need to go to this leaflet. That's why actually these kinds of movement actually happen very low in number. So now the question comes that how scientists discovered that such kinds of movement. How they discovered? Actually, let's see, this is one biological membrane. These are the lipids. Let's see, these are the lipids actually. So they actually attach some dye to these lipids head actually. For example, this is one dye they actually attach. This is one dye. Let's see example for attach. After sometimes, for this is a fluorescent dye. Okay, fluorescent dye, and after sometimes they found that the position of dye is different position. Why? Because the dye is attached to the phospholipid. In this way they found that lipids rotate within the biological membrane. Okay, and this process actually known as fluorescent recovery after photo bleaching. This is actually the process of the techniques by which they actually discover such processes. Okay, thank you. If you like this video, then please hit the like button and subscribe. Okay, thank you.